Okay, picking up where we left off. In general, neutral mutations, naturally evolving proteins gradually accumulate mutations. This is neutral evolution. It doesn't really like negatively or positively impact stuff. We just see that creates some variety. This is an example of that. Take your own time to look at this. I'm not going to go through it too much. Please feel free to pause. If you want, we can bring this up and talk about it in class. Obviously not on Friday, but after that. Okay. Other types of mutations that are important to know. A frameshift mutation occurs when I insert or delete a letter. This means that the whole codon, those 3 letters that I read, it shifts what I'm reading. This is usually very, very negative. It may cause like the organism to die before birth, and it can cause very big mutations as well. A frameshift mutation changes like a whole protein. More about like those examples, feel free to look at this at your own discretion. When we talk about other mutations, chromosomal rearrangement is going to be a huge mutation as well. This can result in the emergence of a new species. There are these 5 different types that I will run through very briefly. So this is why we're talking about this. If you look at the karyotype, or the image of the genes of a human and a chimpanzee, we notice that the great apes have 24 pairs of chromosomes compared to the 23 that humans have. Why is it different? Chromosome number 2 in humans is the result of the fusion of 2 chromosomes. AKA, we had a fusion, letter E, of the chromosomal type that has allowed us to evolve far past the apes. We also, as humans, impact the gene pool. We can basically eliminate things. This is called artificial selection. Basically, we choose certain organisms for certain traits, breed them together to change the organism's gene pool. This wild mustard plant, a brassica, if you are familiar with plant terms, you know that a brassica is cauliflower, kale, cabbage, Brussels sprouts, collard greens, Swiss chard. All of these came from one type of plant. The reason that it did was we chose certain plants to start breeding to make a new species. This happens with like dogs, cats, all sorts of plants. We call it artificial selection, and there are often consequences that are involved in such stiff manipulations. In agriculture, if I'm trying to get a very specific type of plant, I end up compromising on the health of the genes of the other plant. This is why corn as a crop is so susceptible to pests, is because they've chosen it to be very fast growing, to have a lot of kernels, to have very high yields, but that means that other traits that protect them from insects and things like that are very weak. When we talk about antibiotics being applied to a population of microorganisms to treat an infection, they can start to become immune. Basically, we don't realize we are artificially selecting for this, but we do. If you get antibiotics for something, right, they give you a set of pills and they tell you to take them all. You are basically creating resistant bacteria by not taking all of them. Essentially, the resistant microorganisms will start to flourish and reproduce, and then you'll have more bacteria that are resistant to that antibiotic, as opposed to just killing them all off. This increase in antibiotic-resistant bacteria has caused doctors to reduce the number of prescriptions written for antibiotics. Because at this point, about 70 negative bacteria are resistant to some sort of antibiotics. They're called superbugs, or MDR bacteria, multi-drug resistant. This is a very scary thing to think about, and a very real thing. An example of this is MRSA. If you've heard about this, this was just new information when I was in college. It is a bacteria that grows on locker room floors and things like that. It basically will start to cause skin boils and burns and lesions that don't heal, so you have raw open wounds, and they will start attacking the internal organs, so they need to basically amputate the part of the body that has these. reducing or eliminating gene pools. We can also impact gene pools in a very negative way. Oops, okay. We lost our little thing, but we're back. Sorry, guys. Let's just keep puttering through. So, other ways that humans can impact the gene pool, we can basically, if we get rid of a large number of organisms, we will see that the gene pool is directly impacted. We can also see that we can prevent genetic drift or diminish gene flow when we impact the environment in specific ways. We reduce the genetic variety and therefore decrease the chance that the organisms will be able to survive because they don't have as many, there's not as much variability. And we know that decline of variation in any case is decrease in stability. The impacts of sexual reproduction on variation. The good thing, after talking about some of this negative stuff, is that sexual reproduction recombines genes in new ways. This is why you and your siblings probably don't look identical. We make 2 to the 23rd different kinds of gametes. And if you combine that with your partner, you have a whole lot of different choices for what your offspring will look like. The probability that you and your sibling will be identical is like minute to the nth power. Sexual reproduction is like shuffling a deck of cards, and every time you pull one card out, it's a little bit different, and that's because of evolution. That is it for this PowerPoint. You should be prepared for Friday with this. Hopefully it was short and sweet and to the point. Please collect any questions you have. I will address them on Tuesday when I see you. Enjoy your weekend.